This is the core component mock number 1. On the front we've got all the marks added up and some instructions. Hope you read those instructions. And then we have got, if I fold that over like that, you can see this is part of a plant cell. So it's only part and it's a plant cell. So the rest of the plant cell would be around up like that. So some people thought this was lots of cells, it's only part. So when you're thinking of a plant cell, I'm thinking, in my mind I'm thinking of cell wall, thinking of membrane, nucleus, mitochondria, chloroplast, ribosomes, Golgi, and so on. Endoplasmic reticulum rough, endoplasmic reticulum smooth. So those are the things that I'm thinking of as soon as it mentions a plant cell. So when it says identify the structures, those sorts of things might pop into my mind. And so we've got these stack of membranes. Now those are the Golgis. So Golgi body, Golgi apparatus. B, again, if you do enough exam questions, look in enough books, B kind of, we've got outer membrane, and then you can sort of see things sticking in like that. Well, that's foldings of the inner membrane into cristae, so that's the mitochondrion. I'm putting these in the singular. If you said mitochondria, plural, that's absolutely fine as well. They won't penalise you for that. C, this looks like layers of membranes. I can't see any dots on it, so it's not the rough endoplasmic reticulum. It's smooth endoplasmic reticulum. The mark scheme just gives it endoplasmic reticulum. So I'm going to put smooth endoplasmic reticulum. D, right, D is this thing that goes kind of all around there like that, and it goes all the way around the cell there, and that is our very thin, nothing else attached to it, so that's our cell membrane or cell surface membrane or plasma membrane. I'm just going to put cell membrane. E, well, we can see other things there, and then this is another cell with another layer of things. So that's our thick cell wall that we are expecting to see around a plant cell. So you're expecting to see it, and so when you look at a diagram you go, right, that's one of the things I might expect. So cell wall. And F, little dots inside cells. Well, there's only one thing that you know are like little tiny dots that don't appear to be kind of membrane sacs or anything, just little tiny dots. Those are going to be our ribosomes. State the name of 2 organelles or structures which may be found in plant cells, so this one, but not shown. So not shown in this one. Well, here's our, I mentioned it earlier, nucleus. You certainly expect to see a nucleus there. And I can't actually see things that plants are famous for, photosynthesizing, so I can't see any chloroplasts here. I don't think that's obviously a chloroplast. So those are the 2 things that I thought of at the start. You could have had things like starch grains, plasmodesmata, things inside the nucleus like nucleolus, chromatin. We can't actually see any rough endoplasmic reticulum. They're sometimes quite hard to tell, or a large kind of vacuole. But I would those two are the most obvious things that I can't see there, so I'd go with that. Substance P, so this kind of white stuff over here, has been assembled in the Golgi. So it's been assembled here in the Golgi, and it's secreted from the cell. So it's made there, and then it's going out of the cell. So how does that happen? Well, we need to draw a little diagram, so I'm just going to draw a Golgi. So there's our Golgi, and it would then travel by means of vesicles. So we can see some vesicles here. So I'm going to put a little arrow there to show where it's going, and then that would get to our membrane, where this vesicle would kind of fuse with the membrane and then release substance P. Or you might have drawn it in a series of diagrams with the vesicle moving, joining on, and kind of releasing the contents. We're going to label this, so I'm going to label vesicle. This is our cell. I can't spell membrane. The name of this process, exocytosis, and I put showing how things are going to move. So not a brilliant diagram, but enough to get us the marks. So the marks were, vesicles coming from the Golgi for a second mark, showing that these are migrating to the membrane as a mark, showing how the membranes fuse for a mark, and then saying the actual phrase here, exocytosis. If you didn't label your diagram, you could only get a maximum of 2 there. So generally people have done quite well in this question, and there's quite a few people got 11 marks out of 11, which was very good.